What do humans, plants, and mushrooms all have in common?
They are made of cells.
Yes, they are made of cells. So that's one thing. There are others as well. Let me just type it in here. So again, you can copy what's being written here. Made of cells. Obviously, I'm not typing in complete sentences here. Let's see, Matthews, I saw that you wrote something as well here. What did you say that they all have in common, Matthews?
Uh, what I said, they all have chromosomes,
Uh-huh. What else? What was that last word that you wrote?
energy, and they have energy.
Yes. Yes, they all use energy. So we're gonna use energy. Oops. Whoa, whoa, whoa. Use energy. Yes. Yep, yep, yep. What is it that viruses can't do? That's why they have to invade other cells. What is it that they can't do? That's why they have to, yeah, they can't replicate. So living things have the ability to replicate or specifically reproduce. Reproduce. So those are a few of the things that you can have there. They also grow. That's something that somebody else wrote. They grow and respond to environment. So go ahead and copy what I have right here. And I'm going to also put it in the chat. So viruses are not alive. Yes, viruses are not alive. That is right. Viruses are not alive. Oops. So let's check it out. So we have a microscope lab today. So I'm going to go ahead and pull that up. And so when you open the gizmo, it has something called the landscape. And the landscape, if you hover your mouse, it glows yellow. But I want you to click on show all samples. You want to click on this checkbox so that it's easy to find things that you need to explore. We are going to explore the Eladia leaf right here. It lives in, it's like a type of algae thing. um, and it lives in the pond here. So basically, you're going to go back and forth between the landscape to choose a sample. You're like plucking it from the landscape, and then you're putting it under the microscope. If you had Mrs. Dwyer, some of you said, I did microscopes. I'm good. So that's awesome. We're going to review that. So instead of um, looking through a microscope, you have your eyepiece that's up here. You have your magnification. You can control how much you're going to zoom in, basically 40, 100, or 400. It'll change depending on your sample. You see these knobs right here? You kind of turn them when you're looking at something under the microscope. That big knob is called the coarse focus, and the little knob is called the fine focus. So the first thing you want to do when you're trying to get a clear picture is go ahead and do this on your own. Move the coarse focus to the left or to the right. So waterfall chat me right now. If I want to make it clearer, should I move it to the left or move the coarse focus to the right? If I want to make it clearer, to the left or to the right? Type in the chat right now. What should I do? All right. So overwhelmingly, people are saying to the right, and I agree. Once it gets blurry again, oh, it's a little blurry. I'm going to go to the left a little bit, and then I can use the fine focus to make it a little bit clearer. So your goal by the end of the lab today is be able to look at different samples and be able to adjust the focus so that you can see things more clearly. Okay. Um, so how would you describe this? This is the warm up number one. What does that look like to you? How would you describe what you're seeing here? And type in the chat if you'd like to share. What are we looking at? How would you describe it? What's the shape? What's the color? What's the pattern? What do you see? shape, pattern, color. Leia says a scale-like pattern. I agree. I think they look like scales as well. If you agree with Leia, then you can type that in. You want to be more descriptive, add the color, right? What type of shapes? I like Albert adding little pieces of green brick-like scale. So Albert is probably typing for number one, they're little green brick-like scales. Yeah, that's right. Now we're going to magnify 400 times. Hexagonish. Yeah, Camilla, I agree. Hexagonish. I'm not like, you can't quite see the shape. You see that it's not quite a rectangle, is it? Let's click on 400x. Now what do you see? How would you describe that? Okay. So that's number two. Okay. Now what do you see? So your goal is essentially, following the instructions, typing what you see. On the bottom, it has show labels. So sometimes the problem will ask you to show the labels, and I should put on my glasses. No wonder why it's blurry still. Show labels tells you the organelles that are involved, and then showing the scale bars allows you to measure the cell. So let's practice measuring right now. This stage here, if I want, I can move it left and right so I can really get a feel for the length of this cell. So I'm going to put it right there. So answer this question, what is the length of the cell? All right, I'm going to put 10, 20, or 40 micrometers. So micro is this little U with the hook. Okay, it just represents a smaller way of measuring things. Type in the chat right now. Based on this scale bar, what is the length of the entire cell? I'm talking about the entire cell. Remember, this cell is from here to here. What is the best answer? This is the entire cell. This is the entire length of the cell. Yeah, so see, 20 over only goes up to here, so the correct answer is 40. You would be doubling that, okay? So that is one of the questions that you'll encounter. It'll ask you how to measure it, okay? The second thing, if I were to show the labels here, and I were to tell you, hey, what is the length of the nucleus? Okay, there's the nucleus. Let me draw over it in pink here. There's the nucleus. Okay, so choose the best answer. What is the length? Let me type in the chat here. What is the length of the nucleus? 10, 20, or 40 micrometers. Okay, 10, 20, or 40. Yeah, and most of this you're kind of guesstimating, estimating here. I agree with y'all. I would say 10 as well. Okay, because it's sort of halfway. Okay. Is anybody confused as to why the measurement is around 10, right? Because if I look at the scale, it's like the halfway point. It looks like an Among Us character. Oh yeah, a little bit. You can see that. So does anybody have any questions on how to measure? So your goal today is to be able to look at the scale, this little black thing here, and know, okay, that is the length of the cell, or that is the width of the cell. Okay. So that's what part A is. So I've kind of already gone over the parts of a microscope. You don't have to do anything here. It just tells you what it is that you're using. When you're done with the warm up, you're going to move on to activity A, and you're going to, again, you're going to measure. There's examples that show you. And then activity C. Activity C is testing if cells are alive. So everybody click on test for life, okay? Test for life. We're going to try to see if Elodea is a living thing. All living things make ATP. ATP is energy. Y'all remember ATP from 7th grade? It's just energy, okay? The fact that I can breathe and talk to y'all, I have ATP being produced. The second thing that living things make is acid because they undergo respiration. When you're exhaling, you undergo respiration. So this is 7th grade review for y'all because you learned about photosynthesis and respiration. I really love these tests because it easily shows. So you have the positive control, the negative control, and then you have the sample. If the positive control matches the sample, then it has it. The negative control is the test that does not have ATP. In this bottom tray, the negative control does not have acid. Phenol red is just like a chemical. You drop in there and see if it changes colors, it tells you it has this. Okay. So we're going to press play and then see what happens. So it says that if it has ATP, it glows green. All right, answer this. Does the sample have ATP? Yes or no? Type in the chat. All right, lots of yeses. Vince, can you tell me why? How do you know that the sample has ATP? How do you know that? So how do you know it's a yes? How do you know, Vince? You can type in the chat or just say it out loud. Yeah, or say what Matthews just wrote. Thanks for helping out. Can you hear me, Vince? There we go. Yeah, Vince typed it out as well. It's green. Remember, the sample is green and the positive control is green. Therefore, it's a match. Therefore, boom, it has ATP. And it literally, this test, if we did it in class, you'd see it glowing green. It's so, so cool. All right, the next test is phenol red for respiration. Okay, so does the sample here have acid? Does the sample here have acid? Yes or no? Okay, so let me type in the chat here. Does the sample have acid? Yes or no? This is the phenol red test. So what color should it be if it is positive? So kind of use that to answer. So yes or no. And the answer is no. Yeah. What color are you expecting? Camilla, and you can type this in the chat or say it out loud. What color should it be if it was acidic, if it did respire, if it did have acid? What color are we looking for here? Yeah, the same as the positive controls. And what color is that? I would be looking for an orange-ish color. That's right. We're looking for an orange-ish color. Yeah, okay. So this is a no. So I would put an X there. It does not. It does not have it. Okay. And so it will test, even though it doesn't have acid, it's still a living thing. It tests positive for either or. Okay. So your goal is to be able to look at a test and be like, is it positive or is it negative? Does it have ATP? Does it have phenol red? Okay. Let me show you what a positive test looked like. Let's go to the landscape. I'm going to click on E. coli. It lives in the gut of a cow. We're going to press play for E. coli. Does E. coli have acid? Yes or no? Okay, remember what Camilla said, what color we're looking for. And the answer is yes. See how it's that orange color. Okay. Now, another thing you can do is if you want a really clear view, you can turn on and off the light switch as well. You really see it glow, okay, if it's present. Okay. So there's that light switch. You can turn on the lights or you can turn off the lights. The lab will tell you to turn off the lights at some point. All right. So that is the overview. And before I have you get started, we're going to take a nice little brain break.